Medicine Without Cruelty presents Animal Testing Why It Must End Right Now. In this podcast, you will recognize the main reasons why animal testing is outdated and ineffective from both a scientific, ethical, and also a practical perspective. And we'll take you through notable cases throughout history. Animal testing has had disastrous effects on human health. Now, animals do not replicate human biology, and the bigger question should be, why are we still using animals today? Firstly, it poorly predicts human responses. Animal testing has repeatedly shown that it fails to predict human results. According to the National Institutes of Health, around 90 to 95% of drugs that prove safe and effective in animals fail in human clinical trials. These failures are not just due to unexpected side effects, but also because animal physiology differs too greatly from humans. And what's the result? Wasted time, money, and animal lives without any benefit to human health. Furthermore, complex human diseases such as Alzheimer's, cancer, and cardiovascular conditions are poorly modeled in animals. While animals like mice or rats may develop similar diseases, the processes that underlie them are just not the same in humans. For example, rodents have vastly different immune systems and metabolic processes, leading to research that does not apply to humans. That mismatch results in misleading data and costly failures in drug development. Now, another critical failure of animal testing is its inability to account for the vast diversity present within the human population. Humans are wonderfully unique. Our differences range from the color of our eyes, hair, to individual allergies. These variations are part of a defining feature of our species and shape how our bodies respond to treatments. Animals too have their own specific intolerances to plants, foods, and other substances, such as essential oils, for example. What may be safe for humans can have disastrous effects on animals and vice versa, as you'll see through the historical cases. Now, these key differences highlight a fundamental biological variation between species. So, how can we trust critical testing of medicines on animals when their biology fails to reflect our own? Now, zoonotic diseases, so these are diseases which originated in animals and have been transmitted to humans. That's been a devastating reality which we have seen. For example, COVID-19 likely originated in animals and highlights the dangers of cross species transmission of deadly viruses. Now, for example, laboratory animals such as primates can carry zoonotic diseases and pose a severe risk for us all. All animals experience stress in their own way, from mice to macaques, and it ranges from pain, stress, distress, anxiety, and depression. We are depriving them of what is natural and bringing them into a situation that can cause severe changes to their mental and physical health of the animal. Now, psychological effects can involve repetitive pacing, obsessive compulsive disorder, and even self harming, much like behavior you might see in zoos. Physical effects can cause severe changes to the immune system, metabolism, and it could even make them more vulnerable to illness. Some of these effects can potentially be passed down through generations. For example, stress in pregnant lab animals have been shown to alter the genetics of their offspring, completely distorting experimental results. And also, another notable example is macaques, who you can see in the middle picture there, who are wild caught and or who have experienced the stress of laboratory life, can pass on these genetic changes, causing an altered biology in their offspring, in their infants. Now, all of this is strongly likely to influence the reliability of scientific data. It can completely distort our experimental results by the very fact that they are in a laboratory setting in the first place. Now, the technologies that are used for animals are obsolete as well because we have new non animal methods. For example, the lethal dose 50 test, or LD50, and it's designed to measure the lethal dose of a chemical. So you might rightly understand it means the death of animals by the toxic effects of a particular chemical. Now, that's been largely removed because there are cell based assays in many countries. However, they persist in outdated labs. Now, there are so many cases in which animal trials have directly caused devastating harm to human health. Now, thalidomide is something that you may have heard of before. It caused devastating malformations in up to 30,000 infants. It was apparently safety tested in 10 strains of rats, 11 breeds of rabbit, 2 breeds of dog, 3 strains of hamsters, 8 species of primates, and various cats, armadillos, pigs, guinea pigs, and ferrets. Now, later investigations in pregnant animals such as mice, rats, guinea pigs did not reveal malformations in their infants. Vioxx, which is an arthritic drug, caused 320,000 cases of heart attack, stroke, and cases of heart failure worldwide. 140,000 of them were fatal before being pulled in 2004. 6 animal studies in 4 different species showed protective effects against heart disease. Isopurel, which is a potential asthma treatment, caused 3,500 deaths in Britain. It was tested on rats, guinea pigs, dogs, and monkeys at a dose that's extremely high. Can you imagine? It was tested in doses that were too high than what was given to a human, and it still caused death and it was fatal in human patients. Now, TGN 1412 was an autoimmune treatment against an immune disease. Now, a fraction of the dose was given to 6 human volunteers who all fell critically ill within minutes and were left with long-term complications. A BI medication, which is for anxiety and Parkinsonian medication, caused brain hemorrhage and necrosis in 5 human volunteers who again were given a fraction of the dose that was given to dogs. It was found to be completely safe in dogs, and one human volunteer even died from the complications of this drug. Now, hepatitis B treatment caused the death of 5 human volunteers despite being safe in mice, rats, dogs, monkeys, woodchucks. Again, the animals were tolerant of a dose that were hundreds of times higher than in humans. The other issue that I mentioned earlier is being able to replicate complex and chronic, i.e., long term human diseases in animals, is fraught with many, many problems. It means we cannot reliably use animals for uniquely human diseases. For example, the chronic disease cystic fibrosis, which causes pancreatic insufficiency, mice have failed to replicate lung infections and pancreatic blockages that occur in these patients. Now, rats have also been used for Parkinson's disease, and they do not get this disease naturally, and so it's induced by destroying the dopamine producing cells of our brain, and dopamine is our feel good hormone, as you know. Now, it does not replicate that progressive nature of Parkinson's disease because it's something that occurs later in life, and it has complex genetic and environmental causes. And what it means is that the treatments that are effective in these rats are completely ineffective in humans. And finally, Huntington's disease, another neurological disease. There might be mouse models which express the mutation of the Huntington protein, which causes Huntington's disease. However, they don't exhibit the full range of motor, cognitive, and psychiatric symptoms that characterize the disease in humans. Thank you for listening.